Now let's get ready to science. Okay, so we started on cell communication. Um, I'll go over the quiz questions after I stop recording, because I meant to do it before I started, and definitely don't want to go. Well, I guess I can, because they'll watch the video after they take the quiz. So let's go over the quiz questions. The first one was to give the two classes of cell communication. What are they? Local and long distance. All right. The next one, I tried to word it as best as I could without saying local and long distance, so you would understand that I wanted you to give the specific types of cell communication. So let's go with local. What's the specific types of local? Paracrine and synaptic. What chemical messengers does paracrine use? Growth factors. And synaptic uses neurons, right? So those neurotransmitters, right? All right. The next one was long distance. What's that called? And what does it use? Hormones, all right? And I think the next one was, what are the three parts of cell communication? Reception, transduction, and response. And then the last part was to describe a generic pathway. And so I'll just read what you wrote on that one, because when we start describing stuff, we may describe it a little bit differently. It doesn't mean it's necessarily wrong, because you didn't describe it the same way as someone else. So, but that last one, basically what you needed to state was that a ligand would bind to a receptor. The receptor would activate protein, which activates an enzyme, which activates relay proteins. That's the transduction of the signal until it triggers a cellular response. Okay, something along the natures of knowing that it starts with the ligand binding to the receptor and then the transduction of the signal through any series of different types of proteins, until it ultimately ends up with a cellular response. Okay, so just to kind of go through, these are the things that you'll need to know from this section of the chapter, which I don't have my glasses on. Oh my Lord. 3 stages of cell communication: how a receptor protein recognizes signal molecules and starts transduction, how a cell signal is amplified with a phosphorylation cascade. We'll talk about that today. What's an example of a second messenger and its role in the signal transduction pathway? We're going to talk about that today. How a cell responds in the nucleus turns on genes, whereas in the cytoplasm it activates enzymes. I'll talk about that today. And we're going to talk about next class, because I'm not going to do the last one today. The next class will be why apoptosis is important to normal functioning in multicellular organisms. So, yeah. Class, does anybody have any balloons at home that they're willing to bring to school Friday? No? I'll go by Dollar Tree. I'm going to suggest that for Friday you bring a towel maybe, or something, or an old t-shirt or something, because we will be performing apoptosis. And when I do apoptosis, just so you remember it, we have balloons, but some of them have whipped cream in them. And you're using your posterior to perform apoptosis. So unless you want a whipped cream butt, you may or may not have one. If you're lucky enough to get a balloon without whipped cream, then you won't have a whipped cream butt. But unless you want a whipped cream butt, you might want to bring something to put around your waist, just in case. It's just a suggestion. I mean, you might want to walk around with a sticky booty. Who knows? All right. Any questions about Friday? No?
I thought we were doing FRQs.
We are going to go over that. But we're also, that's why the only thing we're doing with this, other than going over that, is the last bullet point, 6, which is apoptosis. It doesn't take that long, but we're also going to do an activity to go along with it. I'm not going to completely cancel my year because of COVID. Some activities I just feel like I still want to do because it's fun for me to watch. That one is definitely fun to watch. Yeah. Mm-hmm. Hey, what's the stuff all over your? Well, I forgot to bring something.
I will not forget.
And I was the unlucky one that got a whipped cream balloon. All right. So anyway, so cells. Let me. I don't like being up front. On this. So y'all just keep looking that way. I'm probably gonna be back here. As long as you can hear my voice, you okay? If I need to go up there to the whiteboard, I will. So don't look at me. To the back. that they receive from the environment and other cells, right? That's cell communication. Signals are most often going to be chemicals. And the same small set of messengers show up in diverse species. So one thing that you'll find about AP biology, there is a theme of unity and diversity. And so even though there's different diverse species throughout the world, what we look for is what do they have in common. And one of the things that different species have in common, which shows the unity in the diversity, is the type of chemical messengers that they use for cells to communicate. We see it in more ancient species. We see it across the kingdoms. Some of the same hormones that we see in animals that trigger an endocrine response, we also see those in plants. Now, it's important to note that when it comes to local signaling with plants, the only thing that we know about with plants, the only thing we really understand is how they use the plasmodesmata, okay? But in animal cells, we know about the paracrine and the synaptic communication as well. But when it comes to plant cells, we know that for long distance, they do use hormones. But for local, we know that they use plasmodesmata and not much else is known. So in order to, because you know that science is big on the evolutionary theory. And so there's always going to be an attempt to make a connection between the different species. And so we look for unity in the diversity of the species. And one of those things that we find that's across species is the chemical messengers used for cell communication. So this is what I was asking you guys about. This is not a lion and a gazelle, this is a tiger, but you know, I went on the PowerPoint that day. All right, so we've already gone through, and most of the stuff at the beginning we've already done. I'm just going through the PowerPoint to get to where I need to get to today. Types of cell signaling are local and long distance. We have your local, which is paracrine, synaptic, and your long distance, which is endocrine. Okay, so you see there we have a secretory vesicle, which is secreting those growth factors. And then the target cells. Now, the key to cell communication, cells have different proteins throughout their cytoplasm and embedded in membranes. A cell cannot respond to a signal unless it has the correct receptor. Cell communication is specific. So just because a growth factor is being secreted in the area of cells does not mean that every cell in the area is going to respond. Only the cells that have the correct receptor can respond to the signal. With synaptic, of course, there's your nervous system, so there's going to be a release of neurotransmitters by the neurons. Those neurotransmitters can cross the gap, and then they can trigger a response in the target cell. And then with long distance, we have the hormones, which uses the bloodstream to actually travel through the body. All right, this is showing the gap junctions in the plasmodesmata in plants. And also we have what's called cell-cell recognition. We see this in animal cells as well, where a target cell can have a specific receptor that a neighboring cell has a protein on it, and those cells are able to recognize each other as being the same types of cells because the proteins actually fit like they. So the key here, and always in biology, is that structure dictates function. If the cell does not have the correct receptor, can it recognize its neighboring cell? No. Okay. So we talked about paracrine, and we talked about the fact that it uses growth factors. And so we talked about synaptic and the fact that it uses the neurotransmitters from the neurons. And we talked about the endocrine, the fact that it uses hormones. Again, look at that very last section. The ability of a cell to respond to a signal depends on whether or not it has a receptor specific to that signal. Yes?
So is that why some people can't feel pain because they don't have a receptor for like?
They don't have receptors, so they don't have the receptors to the neurotransmitters that trigger the response that causes pain. So even though the neurotransmitters are being released, their cells don't receive the signal, and so they don't realize that they're actually doing something that's harming their body. All right. Earl Sutherland. Make sure you know this guy's name. Earl Sutherland is the guy that did his research on how the hormone epinephrine acts on cells. Okay? Epinephrine, now I'm going to describe the epinephrine pathway to you using a G protein coupled reactor instead of RTK, but it can work with the RTK as well. Where's my small one? And so, okay. All right, so. Here comes that wonderful art that you guys know and love so much. All right, so here's my receptor. And since I'm using a G protein, what type of receptor would this be? This is a GPCR, G protein coupled receptor. My G protein is here. And the enzyme in this case is a specific enzyme called adenylyl cyclase. Okay? So, what Earl Sutherland discovered was that epinephrine, and we already know that what's this G protein bonded to right now? GDP, and then once it's activated, it'll be bonded to GTP. Okay, so he discovered that when epinephrine binds to a G protein coupled receptor, which in turn activates the G protein, which in turn activates adenylyl cyclase. Adenylyl cyclase is a specific enzyme that catalyzes ATP being transformed into cyclic AMP. Okay? What is ATP? Adenine triphosphate, right? And adenylyl cyclase catalyzes ATP becoming cyclic AMP. So what's the M for? Mono. So it goes from having 2 phosphate groups attached to having 1 phosphate group attached, and then that phosphate group attaches on the other side. You know how normally the phosphate group is attached to the sugar on one side and it's just free? It attaches another side back to the sugar, so that's why you call it cyclic AMP, okay? So the C stands for cyclic. So adenylyl cyclase activates cyclic AMP. Cyclic AMP, catalyzes ATP becoming cyclic AMP. That's the first response, okay? Cyclic AMP acts as what we call a second messenger. When cyclic AMP is created, cyclic AMP is then going to activate protein kinase A. That's his name. That's a specific protein. Protein kinase A. Protein kinase A will then activate another protein, which activates another one, which ultimately leads to, and I'm trying not to get too far ahead of myself because we're going to go into more, I'm going to leave this here for right now. But this is going to activate other proteins, which will lead to a cellular response. The cellular response is the conversion of glycogen to glucose 1-phosphate. What is glycogen? It's made of glucose, and it's how animal cells store excessive glucose. It's stored in the liver. So once this epinephrine actually triggers the release and conversion of glycogen is broken down into glucose 1-phosphate, and then phosphoglucomutase changes glucose 1-phosphate to glucose 6-phosphate. Where are we going with this? What happens? What process? Cellular respiration. So glucose 6-phosphate then completes glycolysis, and if there's oxygen present in the mitochondria, it's going to complete pyruvate oxidation, Krebs cycle, then oxidative phosphorylation to make tons and tons and tons of ATP.
What's the enzyme that converted glucose 1-phosphate?
phosphoglucomutase. It's kind of like phosphofructomutase converts. So this one is phosphoglucomutase because it's mutating a glucose molecule by moving the phosphate group from the number 1 to the number 6 carbon. Okay? So it's phosphoglucomutase. All right, now, Earl Sutherland did his work, and he's the reason we know that we have reception, transduction, and then a response. Yes?
So would that do the same thing if epinephrine didn't, like, bind to a different, receptor?
If it binds to a different receptor in different cells, it's going to trigger a different response.
Okay.
The reason why I stopped with the protein kinase A is because what happens next with the epinephrine depends on the type of cell it bonded to. Like epinephrine can bind to an RTK in different cells and triggers a different response. But the key is that epinephrine will ultimately end in the activation of adenylyl cyclase, which makes cyclic AMP, which then activates protein kinase A. Okay? I made a list of what I want to talk about on my desk, and I should have put it up here, so I don't have to keep walking back there. Yeah, I should put it up here, so I don't have to should have. All right, now, so we know that the 3 stages of cell signaling are reception, transduction, and response. So here you can see reception, transduction, response. Now, I want to make sure because I showed you this using a G protein coupled receptor. The proteins involved in G protein, the starting proteins are all embedded in the membrane, but that doesn't always have to be the case, okay? Sometimes the receptor can trigger a protein. Once the receptor gets a signal, the next protein can be in the cytoplasm. The key is, in order to start, ligand binds to receptor, receptor activates the next protein. All right? So this is reception, and the receptor can be either a part of the membrane or it can be in the cytoplasm, okay? A receptor can be located as a part of the membrane. It's a protein embedded in the membrane. Some proteins go all the way through, so those are your transmembrane proteins. Some of them are integral proteins. They may be embedded, but they don't go all the way through. So, and then other proteins, other receptors are actually in the cytoplasm. Now, most chemical signals are going to be water soluble. What does that mean about them crossing the plasma membrane? If something is water soluble, can it freely cross the plasma membrane? No. So if the chemical signal is water soluble, it has to attach to a receptor that is in the membrane. There's 3 types: G protein coupled receptors, RTKs, which are receptor tyrosine kinases, and you have your gated ion channels. These are going to use water soluble ligands. If a ligand is hydrophobic, like a steroid hormone, steroid hormones are small, they're hydrophobic, and they can cross the plasma membrane. That was one of the questions on your FRQ. How would a steroid hormone get in? They're small. Oh, and just FYI, because I realized we hadn't done cell communication, I didn't take off whether you got the steroid hormone right or wrong. Okay? I just wanted to see what you read about it. They're small, they can cross the plasma membrane. Those hormones can bind to what's called an intracellular receptor. We'll talk about intracellular receptors in a second. So, your GPCRs are your biggest family. They cost the cell the least amount of energy to activate because they use one G protein. Okay? G proteins are very similar in structure, so it doesn't matter which cell it's in, G proteins are very similar in structure. Alright, so this is showing what we already know, right? G protein coupled receptor, an inactive G protein that's bonded to a, what is it bonded to up there?
Or DP.
DP. All right, once the receptor becomes activated, what happens to the G protein?
It loses the GDP and the GTP bonds.
All right, then what does it do next with step 3?
It activates the enzyme.
It activates the enzyme, and then after the enzyme is activated, it triggers a cellular response. What happens to the G protein?
It loses that GTP and the GDP bonds back to its.
No,
It loses an
the GTP loses a phosphate. It doesn't. That's one of the things I want to make sure we have. I don't want you to have the misconception that the GTP goes away and another GDP bonds. The GTP that's there loses a phosphate group, and when GTP loses a phosphate group, it's automatically what? GDP. Okay, that's easier than dropping the GTP and picking up the GDP. Just drop the phosphate group and you have your GDP. All right. So RTKs. Let's talk about RTKs. RTK stands for receptor tyrosine kinase. Receptor tyrosine kinase. Your RTK can actually trigger multiple signals from one activation, from one reception, okay? That's a big difference between an RTK and a G protein coupled receptor. G protein coupled receptor, you get one reception, you get one trigger, one signal triggered. An RTK, because of its structure, can actually trigger different, multiple responses from being activated from the reception. RTKs, when they malfunction, are usually linked to cancer. A malfunctioning RTK is usually linked to the formation of several different types of cancers. All right, now let's look at it. When you look at the structure of an RTK, I want you to notice that it's first made up of two separate parts. These are two different proteins. A ligand must bind to both, okay? So the reception of an RTK is two steps, okay? First, the ligand binds to both receptors, and when that happens, the receptor forms what's called a dimer. It comes together. So that's the first thing that has to happen. The signaling molecule binds to the receptor, and the two receptors have to come together. And this is called a dimer. It forms a dimer. Now notice what residue is on the outside of each one of those receptors. Tyrosine. This is called an RTK. It's a receptor tyrosine, so the receptor has tyrosine on it. And then kinase. What do kinases do? Move phosphate groups. How many tyrosines do I have? 6. So how many ATP am I going to have to use to fully activate this RTK? 6. I have to use 6 ATP. Now let's look at the energy cost. To activate a G protein coupled receptor, I needed one GTP. But to activate a RTK, it requires 6 ATP. Okay? Very, very costly for the cell. Once it has been fully phosphorylated, it is now active. Now, the beauty in this is that these different phosphorylated tyrosines can phosphorylate different relay proteins. Doesn't have to be the same relay protein. Doesn't even have to be for the same cellular response. So these tyrosines, once they are phosphorylated, can phosphorylate a relay protein that triggers a cellular response. So you can get multiple cellular responses from one pathway starting. Okay? That's the big difference. Wake up, Hank. That's the big difference between RTKs and G protein coupled receptors. Any questions about this with the RTK? No? Remember, there's 2 steps to the activation. What's the first step? Formation of a what? Dimer, which means that they receive the signal and they come together. That's the first step of activation. What's the second step? The phosphorylation of the tyrosines. Okay? So, dimer forms, then the tyrosines can be phosphorylated. Capiche? Alright. Last one. Ligand gated ion channel. Easiest one, trust me. You have a protein, transmembrane protein that has like a gate on it, like a flap. It's closed. A ligand binds to it, the gate opens. Whatever it was holding back when it was closed comes out. Easiest one. Protein, close off. Say for instance it's holding back calcium ions, okay? And calcium ions will be found in the smoothie R, so you'll usually find this in the smoothie R, right? It's holding back calcium ions. A ligand, a signal binds to the protein. It opens and releases the calcium ions into the cytoplasm. The reason that calcium ions are actually kept behind a gated channel, because the change in the concentration of calcium ions in the cytoplasm can trigger several different cell responses. A calcium ion is another type of second messenger. There's two second messengers you guys know about, which is cyclic AMP and calcium ions, okay? Calcium ions are another second messenger. Now, we're getting ready to look at a pathway in a minute. There's no third messengers. Let me make this clear. Your first messenger is always the ligand. The ligand is the first messenger. I can have more than one second messenger. Okay? I may have, the ligand is the first messenger. It may trigger a second messenger. That second messenger can trigger something else that's going to be another second messenger. And that one may trigger something else that's going to be another second messenger. I can have more than one second messenger, but if it's not the ligand, it's always called a second messenger. Doesn't matter if it was another one before it in the same pathway. You with me? All right. Intracellular receptors. Again, what types of ligands can we use with an intracellular receptor? They have to be what? Hydrophobic, because they have to be able to cross the plasma membrane. So they're going to be hydrophobic. Your examples of those, of course, we talked about the steroid hormones, also your thyroid hormones, okay? These, and their job, what an intracellular receptor is usually going to do, an intracellular receptor sits in the cytoplasm, the ligand crosses the plasma membrane, activates and bonds with the intracellular receptor, and that forms what we call a transcription factor. Once the ligand binds to the receptor, the entire complex of ligand and receptor serves as a transcription factor. And what transcription factors do is they turn genes on and off. So intracellular receptors more often than not are going to act in the nucleus. So that transcription factor is going to cross the nuclear membrane, go to the DNA molecule, and determine which genes are going to be turned on or which genes are going to be turned off for transcription. Because in order for a protein to be created, DNA has to first be transcribed to RNA. Then the RNA can be translated as a protein. Okay? So transcription factors can actually control which genes are going to be transcribed. That's why it's called a transcription factor. It crosses the plasma membrane, so it's going to either be a steroid or a thyroid hormone, binds to an intracellular receptor to make a hormone receptor complex, which then serves as a transcription factor, which can turn on a gene to make the messenger RNA, which then is translated into a protein. Understand? Cool beans. Moving on. Transduction. What is transduction? It's the middle part. And so what is it doing? Passing the signal, right? All right, it uses relay proteins. The cascade of molecules is multi-step. By it being multi-step, it provides a way to control the pathway. If something is going wrong, if you'll notice when we talk about all of this, even with glycolysis and everything, it happens in multi-steps. The more steps you do something in, the more control the body has over stop regulating it. Yes?
You say it's a metabolic pathway.
It is.
It is.
Alright, so let's look at typically in a transduction pathway. So there's going to be a conformational change in shape. Usually it results in the change in shape of a protein, right? We talked about that. Most of the time you're going to have what's called a phosphorylation cascade. A phosphorylation cascade. In a phosphorylation cascade, a phosphate group is attached to a protein, a relay protein, to activate it. It activates the next protein, and then it is dephosphorylated, okay? So the key is that the protein gets the phosphate group, it's activated, it activates something else, then it loses the phosphate group. Protein kinases phosphorylate, protein phosphatases dephosphorylate.
What?
Protein kinases is right here. Protein kinases phosphorylate. Protein phosphatases dephosphorylate. Because remember, when the protein receives the phosphate group, it's active. Once it activates the next thing, it has to be deactivated, right? You don't want it to stay active because it's a cascade. What does a cascade mean? I'm passing it on to the next thing, to the next thing, to the next thing, right? So the phosphate group, so you're going to phosphorylate. After it does its thing, it's immediately dephosphorylated. The type of enzyme that phosphorylates is called a protein kinase. We've heard kinase before a million times in here already. The protein that dephosphorylates, we haven't talked about phosphatases before, is a protein phosphatase. Kinases add the phosphate group, phosphatases remove the phosphate group. When a protein is phosphorylated, it's activated. When it's dephosphorylated, it's deactivated. Capiche? All right, let's look at it. So, ligand binds to receptor. Receptor activates a protein. That protein is going to activate the first protein kinase. That protein kinase is going to take a phosphate group from ATP and add it to a second protein kinase. Okay? It phosphorylates it. A protein phosphatase will dephosphorylate the inactive this activates this one. Okay. 1 activates 2, 2 activates 3, 3 activates the relay protein or the protein that's going to trigger the response. In between, so to activate it, you have to add a phosphate group. This is a protein kinase. To deactivate it, a protein phosphatase removes the phosphate group. Do y'all see what's going on with it? No?
Like a long chain of relay.
It's a long chain of relay proteins, and the more relay proteins you have, the more ATP you're going to use. Because at each one of these, it requires an ATP molecule. It starts with the signaling molecule binding to the receptor activating a protein that activates the first protein kinase. What does a kinase do? Phosphorylates. So, the first activated protein kinase then removes a phosphate group from an ATP and adds it to a different protein kinase, activating it. Okay? Once it is activated, it removes a phosphate from an ATP, activates a different protein kinase. Once it has done that, though, it doesn't need to stay active. It needs to be deactivated. And so that's where your protein phosphatase comes into play, because it removes the phosphate group. Because how was it activated in the first place? I added a phosphate group. How do I deactivate it? Take away the phosphate group.
Is that inactive protein kinase? Is it the same thing as a protein phosphatase? Because it's removed, or?
Protein phosphatase is a totally different enzyme. Okay? A protein phosphatase removes a phosphate group from a protein.
Oh, that's what the little
PP. That's what PP is. Protein phosphatase.
So it's the thing. It's what removes them.
Right. It's what removes the phosphate group from a protein.
Okay.
kinases remove it from an ATP and add it to a protein.
So it's still protein kinase, it's just inactive or it's just active or inactive.
There you go.
Okay.
Any other questions? All right. So this is the same thing, just showing it more up close and personal. Second messengers. Second messengers are going to be small. They're going to be non-protein. They're going to be water-soluble molecules or ions, and they can spread throughout the cell. Okay? So, small, non-protein, water-soluble, and they can spread throughout the cell. That's what second messengers are. So, cyclic AMP is a nucleic acid. Calcium ions are ions. They are not proteins. Your second messenger cannot be a protein. If it is something that is sending the signal and it's a protein, it's called a relay protein. It's a part of the transduction pathway. But when something is created or activated that is not a protein, and its job is to send the signal on, that's why we call it a second messenger. If the message is going from protein to protein to protein, that's just a part of that's just a relay protein. But if the message ends up with something that is not a protein, and its job is to continue to send the message, then we call it a second messenger. Got it? All right. So, cyclic AMP, calcium ions are your most common second messengers. There's more, but these are the two I need you to know. Cyclic AMP and calcium ions. This is just showing the formation of cyclic AMP, showing how adenylyl cyclase converts ATP to cyclic AMP by removing two phosphate groups, and then the phosphate group that remains. See, this phosphate group is normally bonded to another phosphate group, but here it bonds back to a carbon in the sugar. So that's why it's called cyclic, because this part is bonded back to the sugar. Okay? Cyclic AMP. Closer look. There you go. Alright, so this is showing what I drew over there on the board. My personal preference is that, my artistic disability works better for me. You heard what I said, right? Artistic disability. So this is epinephrine. It binds to a G protein coupled receptor, which activates the G protein, which activates the enzyme. And in this case, the specific enzyme is adenylyl cyclase. Adenylyl cyclase is going to catalyze the conversion of ATP to cyclic AMP. Cyclic AMP, because it is not a protein, will now act as a second messenger by activating protein kinase A. Are you with me? Protein kinase A can then activate other protein kinases or other proteins, or it may directly trigger a cellular response. Okay. Now, remember what I was talking about? I couldn't remember the name of the disorder. Well, I think we called it the wrong thing, but it's cholera. Cholera is the one where the G protein is made to be, where it can't deactivate, so it continually activates the enzyme that triggers a response. And so the enzyme that's continually being activated is cyclic AMP. It's adenylyl cyclase, which is continuously converting ATP to cyclic AMP, which makes cyclic AMP continue to activate protein kinase A, which triggers the cellular response of the sodium being released, which the water follows, okay? So, this is what I was talking about last class. The modified G protein continually makes the cyclic AMP, causing intestinal cells to secrete large amounts of salt into the intestines. And we know where salt goes, water follows. That's diarrhea. What can you not live without? Water. Your cells need water. Untreated person dies soon from the loss of water and salt, if not treated.
How do you treat that?
Antibiotics, because it's a bacteria that actually that modifies the G protein. And so bacteria can be killed with antibiotics. Yes?
Alright, this might sound like dumb, but so I don't know if this is true or anything, but in shows and all that, when someone's bleeding out, they get real sweaty. is that because you're losing a lot of salt? I mean, not
I honestly don't know.
like because sodium's in the blood and all that.
That's possibly why, but I honestly don't know. I have to look that up. But that makes perfect sense as to why you would, but I honestly don't know. I know that sweat is in the results of the body trying to maintain homeostasis. So something is definitely happening that is causing you to sweat, but I don't know if it's salt or temperature. I don't know what would be, you know. Good observation. All right. Calcium ions are another second messenger. Remember I told you that you only have one second messenger? Calcium ions are the second messenger in a pathway. The first second messenger is IP3 or inositol triphosphate. Let's just look at this one here. This makes more sense. Okay. Again, you see which pathway we're using? The G protein. Most common. Alright. Ligand binds to G protein coupled receptor, G protein coupled receptor activates G protein. G protein activates the enzyme, and this time the enzyme is phospholipase C. There is a molecule that can be bonded because a light is kind of attached, it's like dangling from the cell membrane. And this molecule is called PIP2, phosphoinositol diphosphate. So just worry about the PIP2, you have to know the actual name of it. But anyway, phospholipase C cleaves the bond between this molecule and the part of the molecule that's embedded in the plasma membrane. And the part that's left is called DAG. And I always say it's like, DAG, I couldn't go. Like, you left me stuck in the membrane. Like, I wanted to go too. But anyway, IP3 is your second messenger. Where did I tell you calcium ions are usually stored in the cell? Smooth ER behind a gated channel. The gated channel opens in response to a signal telling it to open. IP3 is that signal. So the IP3 is going to bind to the gated channel that holds the calcium ions in the cell, acting as the second messenger because a ligand is a chemical message. So, question. I thought I heard somebody say, whoa, okay. So once it binds to the ligand-gated channel, the channel opens, the calcium ions are released, and they trigger several different chemical responses in the cytoplasm. The concentration of calcium ions has to be kept low in the cytoplasm until it's needed, because calcium ions trigger several different cellular responses. So that's why they are kept behind a gated channel in the plasma, in the smooth ER. All right, any questions? Yes.
So why is the DAG left behind?
It's like the thing that's basically holding this molecule until it's needed to be released. Because this is the key to opening the gate. So this is think of this as kind of like being the key holder, like up there, and then this is the key that's hanging on it. Okay? And so once this is deactivated, it will bond back to this. Like once it has done its job, once it's released, it's going to bond back to that to make PIP2 all over again.
So it's just there until it gets the protein to activate it?
Right. And so what this phospholipase C does is it catalyzes the breaking of the bond between IP3 and DAG. Yes?
So when we're talking about using these different, I guess, our enzymes like phospholipase C and adenonyl cytopase, it's just dependent on whatever the cell's trying to respond to?
Exactly.
So, okay.
And so, like, for instance, this signal, because a cell has many different proteins embedded, okay? This signal, whatever this chemical signal is telling the G protein to activate phospholipase C. If it was epinephrine, it would be telling the G protein to activate adenylyl cyclase.
So they can have both, it just depends on which.
Yes, they can have both. They can have both enzymes. It depends on the signal that binds, and then the G protein knows which enzyme to activate. Okay. Yes.
So does each ligand have like a certain enzyme that it'll go with?
Yes.
But so say epinephrine bonded with the RTK, it wouldn't be the same as if it bonded. It would be the same enzyme.
It wouldn't be because remember RTK, if epinephrine bond to RTK is going to make it form a dimer, which causes a phosphorylation of the tyrosines, and it's going to be several different relay proteins that can be activated from that. Okay, you can have so one ligand can bond to a different receptor and cause a different response.
There you go.
Okay,
When you have those RTKs, is that kind of when like you have that phosphorylation cascade?
phosphorylation cascade can happen with any pathway.
Oh.
Okay, that's just how signals can be transduced. The purpose of a phosphorylation cascade is to amplify the signal, to make the signal bigger so you get a bigger response.
So would you use those for like muscle contractions and stuff like that?
That would be a good one, yeah, because you want to make it, especially if you're in fight or flight.
Yeah, like explosive.
Yeah.
So would adrenaline be a ligand?
Adrenaline is converted, if I'm not mistaken, I think adrenaline adrenaline triggers the release of epinephrine.
Okay.
That's what, adrenaline is what triggers the release of epinephrine so epinephrine can activate the pathway.
Okay.
Okay, so there's even stuff that's happening before you get to the, so adrenaline releases epinephrine, epinephrine triggers the pathway.
And that's when you get a bunch of like ATP.
That's when you're gonna get a bunch of ATP because at the end of the epinephrine pathway of this one, what's gonna happen is you're gonna get a bunch of glucose 1-phosphate. And so that's the response from epinephrine. We just still don't have ATP, but the glucose 1-phosphate is then gonna be converted to glucose 6-phosphate so that the cells can use that in cellular respiration to ultimately end up with the production of ATP. So these pathways have to work together. So we're just doing like a section of it with this, okay? It's not the end all and it's not the beginning, it's not the end, it's kind of like what's happening in the middle. Alright, on the PowerPoint, I don't think I put the PowerPoint, don't put slides in y'all. These are some videos though. These are actually available, these videos are on your online textbook, so if you wanted to go back and watch them. Yes?
Can you go into Canvas? Remember how you had like the page where it had all the different units?
Mm-hmm.
Can you go back and put those on there?
It should be at the top of the page. The first one, it says, it's a button you can click where it takes you back to all the units. It should be at the very top, unless I changed it for the, I know what you're talking about. I changed it for the exam, and I need to just go back and reactivate that page. I will do that as soon as I sit down. Yep, I know exactly what you're talking about. Alright, so response is carrying out the instructions. The response can be transcribing a gene. It could be to stop transcribing a gene. Again, these slides are in Canvas, and I'm gonna go back and put the page back so y'all can actually see it. Signal could be opening and closing of an ion channel. We'll talk about that. Alright, regulating a response. I call it the seat. Seat. S-E-A-T. You regulate the response with the seat. Okay? Specificity, efficiency, amplification, and termination. The response has to be specific, which means if you don't have the receptor, you cannot respond. It has to be efficient, which means it needs to do exactly what it's supposed to do. There needs to be a way to amplify it to make the signal bigger. And then once it has done what it's supposed to do, I need a way to stop it. So that's the termination. Okay? So C, specificity, efficiency, amplification, termination. S-E-A-T. That is how you regulate the response. The more steps in the pathway, the more chances you have to regulate. Yep.
If you use those like the ATP cascade, would it take longer for the signal to get there? Yeah.
Scaffolding proteins. This is a specific epinephrine pathway I want you guys to know. I'm probably gonna have to do the I'm trying to see if this is my stopping point for today. Termination, yeah, because that's hypothesis. Alright. Scaffolding proteins. We already talked about this, but I'll just show it to you, let you know it's on the slide. How one signal can trigger a different response depending on what it actually activates, okay? So same signal, different protein activated, different receptor activated, different response, or same, is this the right one? No.
Scaffolding protein?
Yeah, I'm getting to scaffolding next. Oh, this one gets a different signal, triggers a different response. So, yeah,
Can it only form multiple responses if it's an RTK?
no. Any receptor, except for the ligand-gated ion channel only opens and closes, okay? GPCR, RTKs can do any of these,
Okay.
okay?
But to form different responses, doesn't it have to be a different chemical signal?
If it's a GPCR, to form different chemical responses, it has to be a different chemical signal. An RTK, one response can trigger different signals. Is that what you were asking
Yes,
me?
ma'am.
Oh, okay, well, yeah, you're right. Yeah, you were right. I misunderstood. All right, let's talk about a scaffolding protein. Scaffolding protein is the cell's alternative to using an RTK. Okay? Scaffolding protein is a protein that can be activated by a receptor, and then it itself can have different relay proteins, different protein kinases that actually trigger different cellular responses. So once it's activated, it activates different responses.
It's like proteins in a protein.
It's like proteins in a protein. And it's like I have the ability to activate 3 different responses, so all I need you to do is activate me. This is the cell's alternative to an RTK. Because you know RTKs are energy costly. It takes 6 ATPs to fully activate an RTK, and if they malfunction, they tend to trigger cancer. So, not like you can go in and replace them, though. That would be nice if you could.
So why doesn't your body just use that all the time?
Well, you know, we are a combination of trial and error. So, in some stuff, and RTKs are good for certain things. And so, yeah. That's a $1 million question, though.
Would it send out all 3 of those proteins?
Yep.
Oh, yeah.
Amplification. We see with the epinephrine pathway, this is a specific pathway that I want you guys to make sure you know. You know that when you have a G protein, you already know the first part. This is the first part right here. Okay? Epinephrine activates G protein activates adenylyl cyclase, which catalyzes ATP to cyclic AMP. Cyclic AMP activates protein kinase A. Protein kinase A is then going to activate your phosphorylase kinase. Phosphorylase kinase is then going to activate the glycogen phosphorylase. It's been a minute since I looked at this one. Glycogen phosphorylase is going to trigger the conversion of glycogen to glucose 1-phosphate. So all you're adding is 2 more enzymes. All you're adding in is 2 more enzymes, which is the phosphorylase kinase and the glycogen phosphorylase. Okay? Phosphorylase kinase, glycogen phosphorylase. But what I want you to notice is, look at the magnitude of the response at each step. 1 epinephrine bound to a receptor, that's 1 molecule. Here, I get 100 molecules that's activated, okay? That 100 becomes what? How many cyclic AMP do I get from that? 10 to the 4th, what's that? All right, and then those activate 100,000, and then those activate 1,000,000. So from 1 signal, I get 1,000,000 active glycogen phosphorylases. How much glycogen does it convert?
Oh, that's
A billion.
So, 1 signal, this is what we mean by amplification, okay? Triggered by 1 epinephrine molecule, the cellular response is 10 to the 8 glucose 1-phosphates. That's a lot. And then each one of these is going to be converted to a glucose molecule that can then be converted to up to 36 ATP. That's a lot of ATP. That's a lot of ATP, right? So that's what we mean by signal magnification, amplification. I'm sorry, not magnification, amplification. I guess they mean the same thing, but.
Isn't that the thing that people use for,
Questions, comments, or concerns? All right, let me go ahead and put the, do it while I'm standing right here.
for when you have an allergic reaction? EpiPen?
Yep, an EpiPen is epinephrine, so it can trigger a reaction.
So
With an EpiPen, you're shooting epinephrine directly into your body.
So how does that help?
Peanut allergy.
Yeah, and you're having a reaction.
how you set up your how does that work?
It's gonna activate those mast cells to get those mast cells going to that area to try to figure out, try to get rid of your immune system gets more active faster, basically. All right, this is not AP Bio. This is Bio. Oh, I'm in student mode. That's why I leave student view. Now, can I go here to AP Biology? And which unit was this?
This is 5.
4.
4 of 8. Okay. Y'all see pages? I need to put my glasses on. I can't see. Do all pages. Unit 4. Stop it. Stop moving. I already can't see. Oh, Perry, would you stop that camera for me? Please.